Cell division is required for an organism to grow, mature, and maintain tissues.
Mitosis part 2. You guys started mitosis, how a cell doubles its DNA and copies into 2 cells, and it's basically cloning. We talked about how you have 46 chromosomes. They all have to copy. Each cell, when you're done, should have 46. When they copy, they look like an X that is just going to get ripped apart. And that's the part I'm going to show you right now. Please note that the cell cycle is all of this. These are phases: interphase, prophase, metaphase, anaphase, telophase, cytokinesis. Mitosis is just this part where we're ripping those doubled chromosomes apart. So we're going to be drawing the phases. This is why you have 2 green papers. We're going to have phase 1, interphase. And we'll be drawing what the cell looks like there. We'll have phase 2, prophase. And we'll be drawing what the cell looks like there. We'll have phase 3, metaphase. We'll be drawing what the cell looks like in that phase. Phase 4, anaphase. I call it Anna the Ripper. The cell's a little bit elongated in this phase. There are some characters in this story. Indiana Jones is in here with his whip. Phase 5, telophase.
Tilo, like television or telephone, but you're in Britain. Tilo, the telephone is ringing.
The cell is starting to pinch in the middle here. That's the worst drawing ever. Yikes. And finally, this last phase, it doesn't really have a number. It's just called cytokinesis. Cyto in Latin means cell. Kinesis, cell cutting. Those are scissors. Okay, we're going to look and see what's happening at each phase. In your face, interphase. This is not part of mitosis because your cell is doing normal cell stuff. It also happens to be where all of your DNA gets copied. Please imagine my hair, right? It's strung out all over the field, individual strands, and I copy it. If you were a hawk flying over the field, you would not be able to see this hair, but it would be being doubled strand by strand and just spread out. You can't see it until we put it in a braid, as I mentioned last time, which won't happen until here. That's a test question. When can you see DNA? Here. When is it doubled? Here. The only reason you can see it here is because it gets put into a braid. Cell does normal cell stuff. It even looks normal. Nucleus, endoplasmic reticulum. In here, like I said, there is little strands of DNA, and it's all being doubled at the time. The molecule itself unzips and brand new pieces get put on, and you get 2 identical copies at this time. When DNA doubles, we want to make sure we're being really, really careful that we don't lose it. So we stick a rubber band to hold the 2 pieces together. The rubber band has a name, a centromere. This is a chromosome unreplicated. This is a chromosome replicated. Unreplicated, replicated. Because you've got 2 of everything, this is what they look like when both have replicated. This is where DNA replicates. Is it in a braid yet? No. I can't really draw it because it's a molecule, but once we actually braid it up, it looks like an X because it's been replicated. It replicates in interphase, we put it in a braid in prophase. Prophase is a big one. DNA coils and condenses and becomes a braid. You can see it. Can you see it in Interphase? No. Can you see it here? Yes, because it's in braids. Just like hair. If you're flying over a field and you're a hawk and I took all that hair and put it into a braid and threw the braid onto the field, you can see the braid on the field versus individual strands. So when we look in the nucleus, and the nucleus at this point, this thing right here, is actually starting to break apart. And it breaks apart for a reason. We have to let the DNA out. We're going to line it up and make sure we pull it apart perfectly and create 2 new nucleuses. But we've got to make sure we're organized. They look like braids at this point. If I was being accurate, I would draw 46 of these. I'm not going to because that's a lot. They have their little belts. They're called centromeres. So for prophase, you need to know that DNA condenses and you can see it. It's already been doubled. And a character shows up. It's Indiana Jones. He looks like a number sign or a hashtag. Hashtag Indiana Jones. There he is. Indy has a real name. And I don't love this name because it's too close to centromere. There's 2 Indiana Jones. There's 2 centrioles.
Move to opposite sides of the cell. These have been floating around in the cell the whole time, and now they're going to move to opposite sides of the cell. So the hashtag, the Indiana Jones, the centrioles,
moves to opposite
sides of the cell.
Maybe you remember seeing this guy when you had to color a cell. And you were like, what is this weird logs? They look like Lincoln logs. Those are the centrioles. And I said we'd talk about it later. Now we're talking about them. I draw them as number signs. That's it for prophase. So these are 2 really big ones. DNA doubles, cell does normal stuff. DNA gets put in a braid where you can see it. The nuclear membrane starts to fall apart. And Indiana Jones shows up, moves to opposite sides of the cell. Maybe we should add
the membrane around the nucleus breaks apart.
All we've done is double the DNA. Remember, our job is to make 2 clones. Double the DNA, put it in a braid. Now we're going to line the braids up in the middle of the field to make sure that we get a copy for each cell. So we're going to get organized in phase 3. Phase 3 is metaphase.
During the mitotic phase, a cell will undergo mitosis to form 2 new nuclei, and then divide to form 2 new individual cells during cytokinesis. Mitosis is the process of dividing the duplicated DNA of a cell into 2 new nuclei. Mitosis is only one part of what is called the cell cycle. For many eukaryotic cells, a cell is duplicated every 24 hours. Most of the life of a cell is spent in interphase. Interphase consists of 3 stages called G1, S, and G2. G1, or gap 1, is the first growth stage of interphase. In G1, the cell grows to nearly its full size and performs many of its specific biochemical functions that aid the organism. Next is the S, or synthesis phase. This is an important stage because it is during the S phase that DNA in the nucleus is replicated. The cell next enters another growth stage called G2, or gap 2. It is during G2 that the cell finishes growing. Mitosis is split into distinct stages. The first stage is prophase. The DNA condenses, organizes, and the classic chromosome structure appears.